Good evening, everyone. Thank you for coming to my talk. If you were excited about my LinkedIn post on me showing you what I have in stabilizing mRNA technologies using an ionic liquid-based approach, this is the place. If you had not seen this on LinkedIn and just saw this out of curiosity, I'm glad for you too. And I hope you're looking forward to other videos where I can maybe go more in depth with this. So to give you a brief overview, this is going to use a molecular dynamic simulation based approach from spectroscopic analysis to figure out how ionic liquids can be the next best alternative in stabilizing mRNA-based technologies. If you want to know more about our paper, like this, how the spectroscopy was described, how the molecular dynamic simulation was performed, as well as other aspects we're not going to cover in this video, you can either scan here for our paper, or you can click the link at the video description, which will lead you to the full paper. Without further ado, let's get started. Okay, so why am I focusing on RNA biotherapeutics today? RNAs are a rising star in the pharmaceutical industry from the COVID-19 pandemic, which is still going on, although it's not labeled as a public health emergency of international concern. The vaccine is still being used to protect us from it as we start to see it to emerge again. At 2019, didn't look like we had much hope, neither at 2020, but in 2021, US FDA finally authorized the mRNA-based vaccine from Pfizer and Moderna, as well as BioNTech. This vaccine is encased in a lipid nanoparticle formulation, which I will talk about near the end of this video. But for our sake, this vaccine was so successful in treating and preventing severe illness and death from COVID-19 infection, it won top first, third, and second place in the top 10 drug sales of 2021-2022. It basically beat out individually all nucleic acid-based therapies like plasmids, protein-based therapies like Paxlovid, as well as small molecule-based therapies like Biktarvy. It's so even more successful to the fact now mRNA vaccines are being developed to prevent cancer and influenza virus infections and pathologies. It sounds great, sounds like a wonder drug. Why haven't we done this before? Why are we struggling to do it now? mRNA is inherently unstable. This is because, unlike DNA, if you know it's deoxyribose, deoxy meaning this 2' hydroxy right next to the phosphate group on the left in the green square, is just a hydrogen. Here it's a hydroxyl, which can actually react with the phosphate backbone and causing it to cleave itself. Although more readily what happens is that water or ribonucleases can come and catalyze this reaction, basically fragmenting the RNA. What does this mean for our vaccine? This basically means if your vaccine is unstable, you'll have less protein to be generated from your vaccine, and less antibodies will be produced. Now, how could this be? Well, we believe and think that it could be due to the topology the vaccine assumes when it's in the formulation. Now, contrary to belief, RNA is not always a linear biomolecule like what you'll see in some cartoons. It can assume 3D topology. This 3D topology can actually protect it from ribonuclease-mediated attack, which in turn, once it reaches a host cell ribosome, can be translated into more protein, which serves as B-cell memory ammunition to when the virus infects you, you'll have more antibodies to respond and prevent severe illness. If your vaccine becomes unwound and single-stranded, like in the case of altered pH, different salt conditions, or temperature increase, you just basically have less protein to be generated because it will become degraded faster. Less antibodies, and if you get infected, you better hope it's not a severe illness. So what have we done to prevent this? Well, right now, the cold chain storage method is the gold standard for stabilizing mRNA. Basically, what is done is the vaccine is stored in conditions between -20 and -80 degrees centigrade. That is a very cold range. If you imagine the temperature you can get down to in like your refrigerator, this is like the Arctic, and then some. Now, this method is good, but it has a couple of glaring flaws. First of all, it's very expensive. If you imagine how expensive it is to keep your house cool using the AC on a hot summer day, imagine keeping these conditions in a warehouse type setting all year round. This means not many countries will have the capability to have these type of facilities and prevent it from getting the vaccine into the arms of patients. The other problem is that the vaccine can actually degrade over its journey. So when the vaccine makes it between the manufacturer to the vaccination center, the temperatures the vaccine can be subjected to can be above -20 centigrade or warmer than that, and it can start to degrade over time. Ideally, what we want to do is throw this vaccine on the shelf and not have to worry about it degrading for days or weeks. If you do it now, it will degrade in hours. So we want a method where we can stabilize a vaccine at room temperature conditions before it goes into use. Ionic liquids may be a potential solution. Why am I proposing this? Well, ionic liquids are basically inexpensive molten salts that are very easy to formulate and synthesize and are not very toxic relative to organic solvents that are traditionally out on the market. Ionic liquids have a lot of potential applications, like they can be used to catalyze a reaction or used as drug vehicles to transport your pharmaceuticals to a drug target that's currently in its infancy stage. It can also be used to extract molecules of interest from a solution. Attractive to us is the ability of, say, imidazolium type ionic liquids to stabilize multiple DNA scaffolds at room temperature. Excuse me for a moment. These videos may play. So basically, the other little thing here is that this is DMIM. Actually, this is OMIM. So that is a mistypo. Now, to kind of highlight on this, our collaborators in Dr. Vaden's lab at Rowan University basically did experimental physical chemical analysis of a DNA strand called PU22 in ionic liquid versus KCl buffer. Basically what they saw was the melting temperature of PU22 and KCl is 80 degrees centigrade, whereas subjecting it to OMIM jacked up the melting temperature to 90 degrees centigrade. Now, what does this mean? Melting temperature is basically the temperature required to separate a folded or double-stranded DNA into its single-stranded form. So like here, in OMIMCL, if the temperature is less, if the temperature is less than 90 degrees centigrade, the DNA is stable, it is folded. But if you go above 90 degrees centigrade, yes, it can start to unfold. So basically, this is 10 degrees shy of water boiling for this DNA to start to unfold at. Why am I focusing on DNA though? Okay, so we use DNA as a stepping stone to our target, which is mRNA and RNA. Contrary to popular belief, RNA is pretty difficult to work with in the experimental setting and the computational setting. Although we are doing that now, we wanted to start from the DNA standpoint. Typically because DNA technologies are more well understood, like plasmids, and they're easier to work with, they are more chemically stable. So in this instance, we're working with a G-quadruplex, which is a unique non-canonical secondary structure of DNA or RNA, characterized by the stacking of 2 or more guanine tetrads in planar formation. This structure is basically stabilized by hydrogen bonding and pi-pi stacking between the guanines, in the layers and between layers, as well as coordination of small metal ions like potassium or sodium into the G4 core. It then assumes this type of fold that it can basically form in your genome, and these quadruplexes can be implicated in, say, gene regulation. However, if they are destabilized, they could allow cancer to progress. Thus, they are an actual target of cancer therapies. However, this potential quadruplex is very thermostable, so it is very easy to work with. And the idea is that stabilizing these types of molecules is very well characterized using small molecules, but during different salt conditions, it's not as well characterized. And this is actually the first time that quadruplexes are being used as a model system to, if you stabilize them well, you could stabilize other biotherapeutic methods too. So, long story short, we can ultimately transfer our knowledge we gained from this experiment from DNA to RNA, because there are RNA G quadruplexes that we are working on now that are very similar in topology and nucleotide length and guanine makeup to this particular quadruplex. Now, why do we want to analyze this using MD simulations? Well, MD simulations give us the ability to probe the detailed interactions between the ionic liquid and the nucleic acid structure at the atomic level. While it can be done with wet lab experimentation, it's very difficult. We know that from the thermal melting experiment that the ionic liquid OMI-MCl can stabilize this quadruplex at 90 degrees centigrade versus 80, but we don't know how. Oh, like for instance, we can see the interaction between an anti-cancer drug and this particular Pu22. We can't really measure that very easily with experimental techniques. We can also analyze this kind of result over time to measure if the ionic liquids interact with the DNA in some way. We can see that through the evolution of these systems. So to give you a brief overhead, as like a guide to figure out how the ionic liquids are stabilizing this quadruplex, we're actually using this anti-cancer drug in blue here as a molecular probe. So, because while not really shown here, Vaden's group actually also measured the binding mode of this potential drug to this DNA and saw that it primarily binds to the top site right here at the 5' terminus. What they also observed is that this drug becomes less bound when you add ionic liquid to it. So we kind of want to see what's happening. Okay, now straight to the results. If you are interested in our methods, you can refer to our paper. We're actually going to measure the ability of 4 ionic liquids. The first 2 right here are the more polar ones. They have a smaller alkyl side chain length. If you observe, the TMP binding becomes displaced. It's still trying to bind to the top region of Pu22, but it's having trouble. If you go longer in alkyl chain length, like these two, you'll notice the TMP binding becomes a lot more difficult. And for our big interest here for OMI-MCl, which is where we gained our thermal melting from, TMP is just not able to really bind there at all. Okay, so this gives us a clue. Now I mapped out the distribution to make it easier for everyone to see. The blue dots are basically the binding regions of TMP, that's our molecular probe, throughout the trajectories. And the red dots represent the cation for the ionic liquid, the EM-ion, BM-ion, HM-ion, and OM-ion. The green spheres are the chloride anion of these ionic liquids. The one for KCl and water is not shown. So basically, experimental evidence agrees that yes, TMP can bind to either the top or the bottom binding mode with a side binding mode as like a transition state between the two. That's discussed more in our paper. I'm not going to really go into that here. What's more important is the fact that TMP binding to these regions becomes more sparse. Very specifically, we're looking at the top region because that is the primary binding mode for this piece of DNA. And we see that this top binding becomes more and more sparse and more spread out as you increase the hydrophobicity of the ionic liquid. So at OMI-MCl, it's just not really binding there at all. And the chloride doesn't seem to really have an effect. I mean, it really shouldn't, because anionic, it would repulse with the phosphate backbone of this nucleic acid. So we focus primarily on the cation in how the IL is binding and stabilizing this U22. Okay, so the juicy parts are on the right, but I'm going to go over the left first for consistency's sake. So this can explain the binding of TMP. So basically what happens is that EM-ion and BM-ion are relatively polar, and the imidazolium portion of this cation, which is positively charged, will interact with the phosphate backbone, which is negatively charged. Now, you can imagine that the TMP will overlay at the top layer of this quadruplex, which is facing towards you, and lay flat there if this ionic liquid was not present. But because it is, it's going to lay a bit askew. Once you get to the two on the right, HM-ion and OM-ion, the alkyl chain length increases to an extent that it can actually start to overlay across the top G4 layer, forming like a hydrophobic barrier. This is explaining why TMP cannot bind here. You could imagine that this could be happening at the bottom site too, but due to the geometry of this Pu22, it's not as open at the bottom as it is at the top. At the top, it's way more open. So we think that TMP binding is less significantly because of the increased hydrophobicity of HM-ion and OM-ion. Now, what does this mean for the bigger picture? And also, is this enough to stabilize like RNA or the vaccine? Well, we ultimately want to get to the golden goal, which is simulate a lipid nanoparticle-like environment, but not quite. So basically, the lipid nanoparticle architecture, I promised I would mention this. It's a relatively complex system where you have about 4 lipid components. You have neutral components. You have things like cholesterol or PEG on the outside, constituting it, like the surface. But then you also have ions on the inside that can sequester and neutralize single mRNA molecules. So what this means is that you basically have a large lipophilic vesicle that utilizes charge systems to neutralize and protect your mRNA from, say, ribonucleases, as well as allowing it to get into your cell, because you have to get through the lipid membrane of your host cell. The problem is that this architecture actually doesn't prevent vaccine degradation at room temperature, and it's pretty unclear why. Now, we know that some ionic liquids can actually form micelles, which is what you see at the top right here. Basically, your hydrophobic tails would point inward, and from the hydrophobic effect and the ordering of waters to reduce the overall surface area of water formation, these hydrophobic layers will pack together into like a sphere. And actually, some ionic liquids can use this method to encapsulate and preserve the overall topology of DNA, which is actually Pu22 here. This is currently under review for publishing, and we believe this is a critical step in stabilizing RNA and mRNA-based therapeutics. We've also confirmed this in the lab, that yes, if your ionic liquids are very hydrophobic and induce a hydrophobic effect, you can stabilize RNA. And we are thinking this is the goal to stabilizing mRNA cheaply. I want to give acknowledgements for making this whole thing possible. So I'm on the left. I helped perform the molecular dynamics simulations and did the post-simulation analysis. Phil was able to also do post-simulation analysis. Dr. Chun Wu is my advisor, advising the whole experiment. Us 3 were involved in the computational section of this work. On the right is Vaden's research team for Alston and Hunter, who were very critical in making sure that these experiments went smoothly, basically stabilizing the DNA and performing out the wet lab experiments, and their advisor, Dr. Timothy Vaden, supervised the work. I hope you really liked this talk and found it interesting. Please be on the lookout for other papers of this work. If you liked this work and want to know more, again, the paper will be at the end of this video in the description. You can also scan the QR code. And if you have any questions, reach out to me by email or put a comment in the section. Thank you so much.